Hello, my name is Fabien van der Meers and I'm R&D Technical Leader at Quality Assistance. Today I'm going to present data on the development and validation of an antibody-drug conjugate cytotoxicity assay. Antibody-drug conjugates, or ADCs, are a class of drugs used in the treatment of different cancers. Unlike chemotherapy, ADCs are designed to only target and kill tumor cells. Structurally, ADCs are complex molecules composed of an antibody linked to a cytotoxic compound through the use of a linker. ADCs therefore combine the targeting capacity of monoclonal antibodies and the capacity to destroy cancer cells of cytotoxic chemotherapy drugs. For example, Kadcyla, also known as trastuzumab emtansine, is an ADC that combines the humanized trastuzumab antibody, an anti-HER2 antibody, and the potent anti-microtubule agent DM1, a derivative on maytansine, using a highly stable linker. This ADC is used as treatment for patients diagnosed with HER2-positive breast cancers. Several tests are required by the regulatory authorities in order to prove the quality, safety, and efficacy of ADCs. Among these tests, a cell-based assay must be developed as potency assay in order to demonstrate the cytotoxic effect of ADCs on target cells, as well as on cells that don't express the antigen. In this context, we have developed a cytotoxicity potency assay using Kadcyla. Practically, target cells will be cultured in the presence of dilution series of Kadcyla. After an incubation period, viability will be measured using a specific viability marker. During the development step, different parameters were optimized. The first parameter was the choice of the cell line. SKBR3 and ZL75, two HER2-positive breast cancer cell lines, were evaluated, and we decided to continue the assay optimization using SKBR3 cell line based on culture characteristics. Cell coating density was also optimized by testing densities from 5,000 to 50,000 cells per well. Based on the cell confluency, optimal condition was a culture of 20,000 cells per well. Kadcyla concentration range was determined in order to obtain an optimal 4 parameters logistic curve with at least 2 concentrations in the upper and lower plateau and 3 concentrations in the linear part of the curve. Concentration from 5.2 to 200 nanogram per ml of Kadcyla using a 1.5-fold change were set as optimal. The next parameter was the incubation time of the cell with Kadcyla. 3 timings were tested: 24, 48, and 72 hours. 72 hours of incubation show the best signal-to-noise ratio and will be used for the following runs. Finally, the type of viability markers was evaluated. Among these markers, WST1 and XTT, two tetrazolium-based markers, were tested. Because reaction time was clearly faster with WST1, this dye was selected. WST1 is a dye that is frequently used as a viability marker. However, WST1 doesn't directly measure the cell viability but measures the metabolic activity of the cells. Indeed, the assay measured the cleavage of the stable tetrazolium salt WST1 to a soluble formation by the succinate tetrazolium reductase system. This enzyme belongs to the respiratory chain of the mitochondria and is only active in metabolically intact cells. If we go to the data, we rapidly observe some issues with WST1 reagent. First, a high variability appears at the upper plateau, as observed in the plot at the right side of the slide. Moreover, a high inter-analysis variability was also detected. If we compare these two graphs, we can observe a strong difference in the optical densities obtained for the upper plateau. Around 1.5 for the first analyst at the left graph, and around 2.5 for the second analyst at the right graph. In these conditions, we were not able to validate the method. The cause of these issues was the fact that metabolic activity is different than viability, especially when cells are in contact with low concentration of ADC. In those wells, cells continue to proliferate and can reach confluency, causing a proliferation stop. Moreover, after 72 hours of culture, some essential nutrients begin to lag in the culture medium. This phenomena cause a decrease of metabolic activity in the cells that enter in latency phase. However, even though these latent cells are alive, they will be considered as dead cells using WST1 revelators. The consequence is a high variability in the upper plateau of the 4-parameter curve, leading to variability in the EC50 value and the presence of a hook effect for low ADC concentrations. The assay was therefore modified using cell-tox-green dye, which specifically measures the cytotoxicity and not the cell metabolism. The cell-tox-green cytotoxicity assay measures the changes in membrane integrity that occur as a result of cell death. The assay system uses a proprietary asymmetric cyanide dye that is excluded from viable cells but preferentially stains the DNA from dead cells. When the dye binds DNA released from cells, its fluorescence properties are substantially enhanced. As observed in the graph at the right side of the slide, viable cells don't produce appreciable increases in fluorescence. However, the fluorescence signal produced by the binding interaction with dead cell DNA is proportional to cytotoxicity. The cell-tox-green dye is non-toxic to cells, and the signal remains constant after exposure of 72 hours, making it ideal for determining toxic effects of treatment. At the bottom of the slide, you can see an example of graph obtained with cell-tox-green, with less variability in the upper and lower plateau. After a new step of development, optimal conditions were a culture of 20,000 SKBR3 cells per well in the presence of Kadcyla, from 5.2 to 200 nanogram per ml with a 1.5-fold change. After 72 hours of culture, revelation was performed using cell-tox-green dye. The validation based on USP 1033 recommendation was performed in terms of specificity, relative accuracy, linearity, and precision. During the validation, Kadcyla was used as sample to assess 5 different simulated relative potency levels: 50, 70, 100, 140, and 200%. For this purpose, 8 runs were performed by 2 different analysts in order to determine the accuracy, precision, and linearity of the method. The design of the run is presented in the table at the right side of the slide. During these runs, each potency level was repeated in the same plate to measure the intraplate precision, in a different plate but in the same run to measure the intra-assay precision, in a different run with the same analyst to measure the inter-assay precision, and finally during different runs performed by another analyst to measure the inter-analyst precision. 2 additional runs were also performed in order to determine the specificity using a HER2 negative cell line or an irrelevant ADC, and the stability indicating power of the method using stressed sample. The relative accuracy was determined by measuring the relative bias for each determination. The mean relative bias and 90% confidence interval were computed for each level using the 2 following formula. The table below presents a summary of the result obtained for the relative accuracy assessment. The acceptance criteria were set as mean relative bias 90% confidence interval must be within plus or minus 20% for potency level from 70 to 140% and must be within plus or minus 30% for potency levels 50% and 200%. As we can see in the table, all acceptance criteria were met for all levels. Precision was studied using Kadcyla at the same 5 different targeted values. The following equations were applied overall and for each individual potency level for repeatability and intermediate precision. The following table presents a summary of the result obtained for precision assessment. Acceptance criteria were set as repeatability must be less than or equal to 20% for all potency levels. Intermediate precision must be less than or equal to 20% for potency level from 70 to 140%, and less than or equal to 30% for potency levels 50% and 200%. Looking at the results, all acceptance criteria were met for both intermediate precision and repeatability. In terms of linearity, graph plotting the log transform calculated potency for each level versus the log transform theoretical potency was created and is presented at the bottom of the slide. The acceptance criteria were set as the slope estimate must be between 0.8 and 1.2. The coefficient of determination must be greater than or equal to 0.96. As observed in the table, all acceptance criteria were met for linearity. The specificity of the bioassay was evaluated in two different ways. First, an irrelevant cell line that doesn't express HER2 receptors was tested. During this test, HeLa cells were cultured in the presence of similar Kadcyla concentrations. The second test used an irrelevant ADC, Adcetris or brentuximab vedotin, targeting CD30 antigen. Adcetris was used at the same concentration in the presence of SKBR3 cells. As observed in the two following graphs, acceptance criteria were set as no signal was detected in the concentration range with HeLa cells or with Adcetris. In order to determine if the method is stability indicating, different stress samples were produced and tested with the method. The following table presents the different accelerated stress conditions applied to Kadcyla, including thermal, pH, oxidative, and UV stress, or freeze-thaw. As shown in the result, the bioassay is able to detect sample degradation. This was mainly observed for pH stress and UV stress. In conclusion, this study demonstrates the successful development and validation of a cytotoxicity bioassay using Kadcyla as a model of antibody-drug conjugate. This study also highlights the importance of the revelator agent choice. Indeed, agents measuring the metabolic activity of the cells are not always the best choice depending on the cell culture conditions. It is therefore more appropriate to use agents that directly measure the cytotoxicity based on exclusion dyes, DNA intercalent agents, or change of the mitochondrial membrane potential. The method was finally validated according to USP 1033 guideline in terms of specificity, linearity, precision, and relative accuracy. Moreover, the method was demonstrated to be stability indicating and therefore allows the detection of degraded sample. Finally, if you need more information on the development and validation of this bioassay, an application note is available and can be downloaded on our website.